Types of hormones. Hormones can be broadly categorized into three main types based on their chemical structure: peptides, amines, and steroids. 1. Peptide hormones. Structure: chains of amino acids. Examples: insulin, growth hormone and antidiuretic hormone. Synthesis: synthesized as pre-prohormones in the rough endoplasmic reticulum, processed to prohormones, and then cleaved into active hormones in the Golgi apparatus before being stored in secretory vesicles. 2. Amine hormones. Structure: derived from single amino acids, mainly tyrosine and tryptophan. Examples: thyroid hormones, T3 and T4, catecholamines, epinephrine and norepinephrine, and melatonin. Synthesis: synthesized from amino acids by enzymatic reactions. For example, thyroid hormones are produced by the iodination and coupling of tyrosine residues within the thyroid gland. 3. Steroid hormones. Structure: derived from cholesterol and have a characteristic four-ring structure. Examples: cortisol, aldosterone, estrogen, progesterone, and testosterone. Synthesis: synthesized from cholesterol in the smooth endoplasmic reticulum and mitochondria. Enzymatic reactions modify cholesterol to produce different steroid hormones. Synthesis of hormones. Peptide hormones. Gene transcription and translation. Genes encoding peptide hormones are transcribed into messenger ribonucleic acid, which is then translated into preprohormones. Post-translational modifications. Preprohormones undergo cleavage to form prohormones, which are further processed into active hormones in the Golgi apparatus. Storage and release. The active hormones are stored in secretory vesicles and released by exocytosis in response to specific stimuli. Amine hormones. Enzymatic conversion. Amino acids like tyrosine and tryptophan are converted into hormones through specific enzymatic pathways. Thyroid hormones. Thyroid hormones are synthesized by iodination of tyrosine residues in thyroglobulin, followed by coupling and release of T3 and T4. Steroid hormones. Cholesterol precursor. Cholesterol is the precursor for all steroid hormones. Enzymatic pathways. Enzymes in the mitochondria and smooth endoplasmic reticulum convert cholesterol into various steroid hormones through a series of biochemical steps. Mechanisms of action: hormones exert their effects through specific receptors, which can be located on the cell surface or within the cell. Peptide hormones. Membrane-bound receptors. Peptide hormones bind to receptors on the cell surface, triggering a signal transduction pathway. Second messengers. The binding of the hormone activates second messengers such as cyclic adenosine monophosphate, inositol trisphosphate, and diacylglycerol, which mediate the cellular response. Example: insulin binds to its receptor, activating a cascade that increases glucose uptake by cells. Amine hormones. Membrane-bound and intracellular receptors. Catecholamines bind to surface receptors, while thyroid hormones bind to intracellular receptors. Signal transduction. Catecholamines activate second messengers, while thyroid hormones influence gene expression directly by binding to nuclear receptors. Example: epinephrine binds to beta-adrenergic receptors, increasing heart rate and force of contraction. Steroid hormones. Intracellular receptors. Steroid hormones pass through the cell membrane and bind to receptors in the cytoplasm or nucleus. Gene regulation. The hormone receptor complex binds to DNA, altering gene transcription and protein synthesis. Example: cortisol binds to glucocorticoid receptors, modulating the expression of genes involved in glucose metabolism and immune response.